Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
go above and beyond.
They become very good at what they do, but only
0.1%
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
with Richard Jacobs.
Hello, this is Richard Jacobs with the Finding Genius Podcast. My guest today is Dr. Daniel Geschwind. He's the Gordon and Virginia MacDonald Distinguished Professor, Senior Associate Dean, and Associate Vice Chancellor of Precision Health. He's based out of UCLA. We're going to talk about what's called exome sequencing. So, Dan, thanks for coming.
Thank you, Richard. It's my pleasure.
You know, I think people understand DNA, but what are exomes? What part of DNA is that? What do they do?
Sure. The exome is the approximately, let's call it 2 to 3% of the entire genome that actually codes for protein sequence. So from the exome, RNA is made, and from the RNA, the proteins are made. And so the exome is the part that directly codes for proteins. And the rest of the genome is involved in the regulation of those sequences, that is, turning them on and off, et cetera.
So the exomes code for the RNA that assembles proteins at the ribosome, or more specifically?
Yeah. So the exome is the part of the, what happens. It's interesting because when the RNA is made, it's made so basically every gene, let's call it a gene for a particular protein. Let's take hemoglobin, for example, which is in your red blood cells and carries oxygen. So the gene for that consists of parts that code for protein and parts that involve the regulation of the gene, its levels, turning it on and off. And so from DNA, RNA is made, and then protein is made. And what happens is, in the first case, when RNA is copied from the DNA, it actually includes virtually the entire gene, including what are called the introns, which are the regions between exons, and the exons together make up the coding sequence. And then those regions are spliced out so that you get a message RNA that then gets attached to the ribosomes, and that's where protein is made from that. And it's the message RNA components for all genes that's the exome. In genomics, we use, we often end things with ome. And so a gene is a singular gene. A genome is your entire genome. In the same way, each gene is made up of exons and introns, but the whole complement of exons making up the protein coding sequences of all genes is called the exome. So we're sequencing that ome, that 2 to 3% of the genome that codes for proteins.
Okay. So a gene is composed of an exon and an intron. The intron part governs access to the exon part?
Yeah, it's unclear what, a lot of the genome has unknown function, but we consider introns, which are, so like when a gene starts, it has an area called a promoter, which is before the coding sequence. Then it has, let's say, an exon, then an intron, then an exon, then an intron, et cetera. Some genes have like 40 or 50 exons and introns. Others, there are a few genes with no introns. It's just one big exon. But most genes have introns and exons, on average, 8 to 10 or so. And so the non-coding, which means non-protein coding part of genes and the genome, are thought to be regulatory. Only 5% of that regulatory sequence is well understood. The rest of it isn't that well understood yet. So it's a really interesting point that you make, is that a lot of intronic sequence function is not well known. And that's actually one of the reasons why we don't spend a lot of time analyzing it, and why we actually go for the exons or the exome, because we totally understand their function. We know that they're used to make protein, and certain kinds of mutations will disrupt that process of making protein, and those are usually disease-causing mutations.
When you have epigenetic marks like methylation and histone modification, does that affect both exons and introns or just exons?
No, it affects epigenetic modification affects all of it. It can affect exons and introns, but again, epigenetic modification usually leads to changes not in the protein coding sequence itself, but in the regulation of that gene, whether it's turned on or turned off in a particular cell or tissue, at what stage, and at how much level.
Have people been able to discern if you have a gene with very few introns and very few exons, and it's epigenetically marked versus one that has, like you said, 40 or 50 exons and introns, can you tell if there's a difference in gene regulation? Are ones that have less features easier to regulate or harder to regulate, or is there more nuance to the regulation?
That's what it's thought, that those are more complicated regulation. And indeed, that's the case, in general. I would say there's a general trend towards that. When you get to any specific case, you have to be a little careful. But in general, that's exactly right. The more kind of introns, the more exon introns, the more complex a gene's regulation usually is.
So when someone says, their genes have been sequenced, how much of someone's genome has been sequenced? Like the most sequenced person on the planet, have they gotten everything, or is there still missing pieces?
No, there's still missing pieces, although, we probably have at this point, a good part of it, right? When the first human genomes were done, the first complete human genome was probably about 60%, but it was enough to provide, you could imagine, the analogy might be a building. You see the framing going up, and having that framing tells you how big all the rooms are going to be, where they are, where the stairs are going to be, it gives you everything. And so you could say we had 60% finished, which is like having, equivalent to having the framing. But very detailed framing, maybe with some wiring and electricity and other stuff in there. And now we're beyond that, but there still are regions that aren't well captured. And that's because they contain a lot of repetitive sequence, for the most part. And and part of it is because, and there are also some complex rearrangements that go on in our genomes. And it's because the dominant technology that's been used to sequence genomes uses short reads of DNA, let's call them 100 to 150 bases, and then assembles the genome from that. And if you have long repetitive stretches or stretches that are similar to other stretches, it makes it very hard to map those. And so the advent of a long read sequencing has now made it possible to fill that in. But that's not clinically used yet. So people use short read sequencing, and the vast majority of people who have had sequencing have had their exome sequenced because, frankly, that's the part of the genome that's most interpretable. So you, in some ways, get your most bang for your buck. I'll give you an example if that's all right. Or do you have a...
Well, quick question, then the example, please. So today, the most sequenced person or people on Earth, how much of their DNA has been sequenced? What percentage are we up to now?
I mean, that's hard to say. The most sequenced person on Earth. That's a tough one. I would say it's somewhere in the 90s.
Okay.
Yeah. It's pretty close. Yeah.
One more question here. So what if I had myself like sequenced to the max, as high a resolution as we can get, 90 some odd %, and I did it, every month for a year? Would you expect to see many differences or would you expect to see any differences?
No, I would not expect to see differences. Now, here's an interesting thing. That is, I might see differences if I'm sequencing certain cells that change over time. For example, your T and B cells, which have areas that recombine as you're exposed to antigens and things. And so your immune system genes the genomes in those cells change. But most of your somatic cells, that is, most of the cells in your body, don't change. Over time, there could be some mistakes made as your DNA mutates and then gets repaired.
Yeah, I would think there'll be a base rate of change, and from what I
there
understand,
is,
there's
but
transposons and stuff that change the DNA. But
yeah, but that's not going to really. That doesn't change your sequence from in general. It so modest as to be really hard to assess. Where it would change is sometimes people get mutations that cause cancer, right? So in cancer cells, certainly you're going to have changes relative to your kind of germline, DNA that you're kind of born with. But in general, the kind of rate of somatic mutations, like if I take a tissue, let's say like brain or liver, it's very, very small. They are occurring, but I doubt I would even detect them in most whole tissue sequencing unless one did a study that's very, very specifically focused on detecting those, which requires very deep sequencing and kind of specialized analysis. So in general, that's kind of below the threshold of detection because we're talking about detecting something that's happening in 1% or less of your genome.
yeah, if I had like unlimited money and I said, hey, when I die, I want you guys to deep sequence as deep as possible 100 different cell types in my body. Do you think that would reveal anything useful or interesting? And
yes,
would that be like unbelievably expensive and it hasn't been done?
Yeah, it would be unbelievable. It would be very expensive. It hasn't been done, but people are beginning to work on this issue because we need to catalog and understand what is the extent of this so-called somatic mutation, things that you accumulate over time, and could those possibly contribute to disease? There's quite a bit of controversy around that. And so the kind of sequencing that we're talking about doing clinically for patients right now has to do with taking blood for the most part. Sometimes they're tissue, but usually blood, and sequencing that tissue, not specific cell types. So we're getting a kind of composite view, which is a largely 99.99% accurate view of their sequence and genome. And we're identifying the areas that we can interpret. Now, it's true that what I was going to add before is that when we sequence this 3 to 4% of the genome, the exome, in patients clinically, let's say at a place like UCLA, where patients with a wide variety of undiagnosed disorders, typically rare, are referred. It could be anything from a cardiac disease, an immunologic disorder, to autism or a brain disorder, or another type of neurodevelopmental or developmental disorder. We have a yield of almost just somewhere around 25%. It's a pretty high yield. That is, in people who have not been sequenced, who have a disorder that's been undiagnosed, that have gone to a bunch of doctors, there's a yield of about 25%. If we add whole genome sequencing on top of that, we, let's say, roughly double or triple the cost, but we don't double or triple the yield. And that has to do with a lot of technical factors. And so that's why currently, clinically, most discovery starts with, and clinical diagnosis starts with exome sequencing as a first line.
So, one more question here, and then I promise we'll get to the benefits of exome sequencing. If again, an imaginary study where now I had, 20 different people who all agreed upon death that 100 different of their cell types each would be sequenced to the max, and then you looked amongst them, would you see much difference then? Or do we know? Are there certain cell types that, are almost the exact same between people and some that tend to be very divergent?
That's a great question. It's just not known at all. That kind of study hasn't been done, but the sense would be that there's cells like brain cells and muscle cells that don't divide very much. I mean, there are some certain specific brain cells do, but vast majority of brain cells, neurons, don't divide, of the neurons in the brain. And so cells that proliferate, like most of the other cells in your body, your skin cells, your gut cells, liver cells, etc. It's interesting how those might change a little bit over time with mutations. You can imagine if you actually got a mutation that had some kind of proliferation advantage that made that cell stronger, more fit, whatever, it would overtake the others. And in some cases, that's what leads to malignancies, cancer.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
So now we'll go back to exome sequencing. So I see why it's like, I don't call it low-hanging fruit, but like you said, it's the most well-understood part of our DNA. It appears, the function of it. So what kind of exome sequencing are you working on, and what kind of outcomes are you looking for?
Sure. I mean, you might ask, like, why would you sequence 4% of the genome when you can sequence, 95% or 90%, right? And which is a great question. And up until now, the majority of the answer to that has been the two things. One that you brought up, it's low-hanging fruit in that we can interpret it. It's easier to interpret. And two, cost. So when you look at cost benefit, it actually has a huge advantage. But as genome sequencing cost goes down, it will replace exome sequencing, no doubt, because when you sequence the genome, you get the exome plus you get other things. And we're beginning to be able to interpret those other things more and more. And as we get better and better at that, the value of genome sequencing clinically, its actual output will clinically relevant output will also increase. And so exactly where that inflection point is not clear, but roughly a genome costs about, roughly about $600 and something now to produce using the common technology. An exome around $200, so it's about a third. The exome is 10 to 20 times faster to analyze, at least. So it's kind of an easier process. And so once the cost of a genome gets to be in the $100 range and the computer costs aren't crazy, I believe we're not going to be bothering with exomes anymore. And I think we're rapidly moving towards that place, whether it's $100 or $150 or $200 or $75 is a kind of more of an economic question that will have to be answered. And, but so in some cases so for example, so we start with exome sequencing. It's so inexpensive, and it's actually been in clinical use for not quite a decade, but almost a decade, about 8 or 9 years at UCLA as a clinical test for patients who come in with an undiagnosed disease. Again, as I mentioned before, many of these patients have gone through a relatively arduous diagnostic odyssey. That is, they've spent years, often if it's a child, their family has gone to a half dozen different expert physicians around the country. They've had tens of thousands of dollars worth of tests without any yield. The exome sequencing, which involves sequencing the parents and the child to compare their genomes and see if the child has any new mutations, which is typically the lowest hanging fruit.
What is the highest level of sequencing that I could get right now that would be useful to me, that would tell me something that is understood and known? And what would that be? What would it tell me? So
the question is, it really depends on what your situation is. If you're somebody who has a whole range of neurologic or cardiology or other, or family history of those things, it might be quite valuable. The issue is that we don't do that clinically in patients for whom we don't have a treatment. And so I'll kind of loop back to that, because I think your question kind of jumps ahead a few, not light years, but a few, 100 feet ahead of where my head was at as we were talking about this, because the clinical exome sequencing as a clinical test is extraordinarily useful. So in people who have autism, in a serious developmental delay, serious epilepsies, serious cardiac diseases from arrhythmias to cardiomyopathy, people with lipid disorders like high cholesterol, etc. These rare immunologic disorders, the yields can be incredibly high, up to 50% in those patients.
What does that mean, the yield?
That means that somebody has gone around to see, I'm just going to give you an example, like this diagnostic odyssey. A patient is like 5 years old. They've had a number of developmental delays and problems. They've seen 6 doctors. They've gotten MRIs, EEGs. They've been tested up the wazoo. Let's call it $10,000 or $20,000, down the road, and years of searching. They come to a place like UCLA. They get a clinical exome test in our undiagnosed disease center that's called the California Center for Rare Diseases. And for, a couple $1,000 because we sequence it, because it's a clinical test now where it has to be interpreted, and we also sequence the parents. We end up making a definitive diagnosis in about 25 or 30% of cases. That's the highest yield clinical test we have in general. It's kind of amazing. And so the point is that's the kind of clinical, and the other clinical implementation now, they don't sequence yet the whole exome, but a lot of the exome they sequence in several hundred genes in people who have cancer now. You probably see these Foundation Medicine advertisements or other things on TV. About 6% of people with cancer have a mutation that can currently be identified and lead to a change in treatment where you get a specific drug targeted at that mutation. And that number is going to grow and grow and grow till I predict more than half people with cancer will have targeted treatments. Probably, eventually everybody.
That's great.
So clinically, those are the primary areas where this kind of sequencing a lot of the genome or the exome or a huge panel of genes is being done. In people with familial cases of dementia, like Alzheimer's or frontotemporal dementia and other things, it also has a yield. People with certain adult onset neurologic disorders as well.
Do you think it's worth it for, if someone has the money? How much would it be for me to sequence myself to the max, to get to that 95% or maybe higher level? And, would that be useful, even not now, but, if I hold on to that data for, obviously 10, 20 years, do you think it might be a good idea to do that now? And then as more, as the ability to analyze it improves it, I can then avail myself using what I have to see what that tells me.
No, Richard, that's a wonderful question. I think that is a kind of choice that one can make, but let me spell out the pros and cons in that for you. So, and it really depends upon how great your curiosity is and how big your pocketbook is. So,
well, at least my imagination is as big as you want. So let's talk.
Yeah. And so the issue is that right now, let's say for cancer, we get about 6-7% of patients, and 5 or 10 years, it's probably going to be something like 50%. It's hard to know exactly how fast things will go. And the same thing will occur in other spheres. We'll be able to use a lot of genetics to predict your potential risk for things. Not in the way that 23andMe does it, which is using common genetic variation and generally, in most cases, fairly weak. And as they say, non-definitive. It's not a medical test, right? It's more, for curiosity. My bald brother has a 0 risk, according to 23andMe, of having male pattern baldness, for example, and a bunch of other things. You get the idea. But the idea is that once it gets cheap enough, these data will have extraordinary medical utility because, as I think you alluded to, once we have genetic sequence from hundreds of thousands or millions of patients with a wide variety of common disorders, we'll have the power to understand which genetic variants have the largest effect in causing or increasing your susceptibility to that disorder. So the kind of larger the sample size gets, the more predictive power we get up to a point. But that point hasn't been even reached at all, as in medical centers, really only on the order of hundreds of thousands of people have been sequenced. There's a UK biobank study. And really, in some ways, this pharmaceutical company, Regeneron, has been leading in that way, in that they've partnered with the UK Biobank and with other medical centers, starting with Geisinger, and now most recently with us, to collaborate to sequence a large number of patients. The value of that, along with the medical record, is that now you can ask, how does genetic variation increase risk for certain diseases? So you can discover new disorders, new relationships between mutations and disease that help you understand what we call disease pathophysiology, or the cause of the disease and its mechanism, so that we can design targeted therapeutics. And and we're really in a realm now where we can do that using genetic engineering, as everybody has seen from these rapid deployment of vaccines, using a lot of the same technology that would be used, to approach disease. And so the idea here is that we need to, in certain cases, the yield is high, right? And my belief is that eventually, and whether this is 2 or 3 years again, or 7 or 8 years or 10 years, insurance companies or your healthcare systems, your hospitals, will just get your genome sequence, when you're born, or, in every patient when it's cheap enough, because the value of having that information in terms of being able to predict who is at highest risk for certain cancers, who is at risk for heart disease, who is at risk for diabetes, and actually preventing or intervening to delay the onset or the severity of the condition is going to be massive. And I think that's really where we're going. And for example, in this project with Regeneron that we're starting, that I think sparked you reaching out to me, in sequencing 150,000 patients, we expect that 2 to 3% of those patients are going to have mutations in about 70 genes that are known, that are medically known, that cause medically known syndromes or conditions, and for which there is a kind of treatment. And what I mean by that, the treatment doesn't always have to be curative. It could be, we know this person is at really high risk for colon cancer. Starting at age 25, they're going to get colonoscopies every year so we can detect it early and take them out, right?
Right, it makes sense.
Until we have a better treatment. Same thing with breast cancer, BRCA1, 2, people, but up to about 3% of patients are going to have those mutations. And it doesn't just have implications for them. It has implications for their first degree family members as well, like their offspring and brothers, sisters, etc. So the point is that by knowing that before people reach the age of risk, you can implement preventive procedures that will likely substantially reduce the impact of that mutation on that patient's life and allow them to kind of take control, along with their health system, of that predisposition that they had but didn't know about. And so,
what about epigenetic marks? Can you get an idea of all the marks that affect, at least the exome of a person? And is that called something? And is that clinically useful yet?
No, it's not clinically useful yet, but I also believe that will become clinically useful as well, especially because many epigenetic marks really reflect environment, and they change with environment and exposures, food, exercise, other things, right? And even drugs that you might take. And so they're kind of, once we understand them better, they could be surrogates for environmental exposures and kind of be used as a composite for understanding that risk. Epigenetic markers may be very useful as biomarkers for predicting risk as well, because as you're noting, not everything is in the genes. And let me make a clear point. It could be that for the majority of patients that we sequence, more than 50%, let's just say at UCLA we have 4 million patients, and we sequence them all. I would say for probably 3 million of those, at least, I would agree with this statement. It's unlikely that this sequencing is going to identify rare mutations that are driving disease in them. But for the 5% or 10% of patients that we actually do identify those in, the life savings, the years saved, the pain and suffering saved by the patient, as well as the savings and cost to the public health and health infrastructure is likely to more than warrant that level of screening. And that's why I kept saying as prices drop, eventually this will be implemented, because it's not going to be predictive in the way that I've been describing in everybody, but about 7 or 8% of people likely have rare mutations that either cause rare forms of common diseases or rare disorders. That is, when I'm speaking about rare disorders, they're individually rare, but together they might comprise as much as 10% of our patients.
Okay. Well, very good. Well, Daniel, what's the best way for people to find out more about Regeneron's work and your work specifically? Where can they go?
Yeah. So this was a project that is driven by UCLA Health and our Institute for Precision Health. The notion, everything we've been talking about, really derives from the notion that we can use big data and genomics to care for the individual as the individual that they are. And as you mentioned, their genome is part of that, their exome, and their epigenome. And the more detail that we get from the individual, the more data we have, the better we're going to be able to treat that specific individual and therefore the entire population. And so we have an Institute for Precision Health that's been driving our biobanking, this collaboration with Regeneron, a lot of the implementation of big data into medicine, along with our computer science colleagues. And so if you go to the Institute for Precision Health website at UCLA, we have information on our various programs, which include the kind of e-health, the electronic health, the connection of electronic devices to health record. It involves this population health project of sequencing 150,000 or more patients called the Atlas Project. We have the California Center for Rare Diseases, as well as projects that we're beginning to spin up in cancer and other areas, so that we're treating people as individuals and we're able to implement genomic medicine, which is really the future of medicine, using these omic technologies, not only to diagnose patients, but also in the end to treat them as well. And again, the COVID vaccines are great examples of novel omic technologies that 5 years ago might not have been possible.
Very good. Well, Daniel, thank you for coming on the podcast. It's been a great call, and I really appreciate it.
It's been a pleasure. Great questions.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.